All right, biology. I'm looking back at what we did in class that last day, whether it's Friday or Tuesday, whenever you're watching this. We know that we covered about the cell membrane. Just want to cover that real quick. We know it's selectively permeable. It allows some things to come in, some things to go out. It disperses them accordingly. So selectively permeable. Then we can see it's a phospholipid bilayer. We see these little phospholipids with the heads and then the tails right here. It's the two-layered bilayer. Okay, it makes up two layers. This cell membrane. This completely surrounds the cell. These bodies go towards water. These are the heads want to be attracted to water. So whether that's outside the cell or inside the cell or vice versa, right here. And then the tails are hydrophobic, so they don't go to it. And we saw that there's little things sprinkled throughout the cell membrane. All right, we're going to look some more at these channels as we move forward, these proteins that oftentimes make up these channels. All right, we talked about the fluid mosaic model. And then we talked about how things flow from a higher concentration to a lower concentration, whether we're talking about certain carbohydrates, different types of molecules, or even water. Okay, passive transport, remember, requires zero energy. Okay, it just flows from high concentration to low concentration. Just like an hourglass, let's say. It moves from where there's a bunch of sand in it to where there's less sand. Okay, of course, that's gravity helping it do that. But kind of the same principle. It moves from a higher concentration to lower concentration, and no energy is required for things to move like that. Diffusion is when it moves from that higher concentration to lower concentration. We talked about concentration gradient, where it's trying to equalize both sides. We're trying to get equal value or equal volume on each side of it. Carbon dioxide, oxygen, it tries to do those. We looked at this little thing about which one's moving which way. Obviously, these red, some of them's going to move over here to try to equalize. Same thing with these little blue. They're going to try to equalize. Here, there's going to be some blue coming over here and some red. We're going to see some red coming over. This is fairly stabilized to be more equal. It's going to probably move one of these molecules over, so that way both sides of this gradient is equal. Same principle here. Well, we move like 1.5-ish, trying to equalize. And here we've got about the same amount of blues, but we'd move 1 or 2 reds over, okay, to meet that equilibrium. Osmosis is simply the diffusion of water. It's the movement of water from a higher concentration to a lower concentration of water through a semi-permeable membrane. Okay, so just know that osmosis is nothing but diffusion of water. I told you from the beginning, water is special. So it even has a special term here. All right, let's look at this. We probably didn't get to this, so let's cover it. When there's a difference in water, this is our cell. These little red things are our cells. And then everything outside here is the fluid surrounding the cell. When there's a different concentration of water, certain things happen, okay? In this first situation here, this is an isotonic solution. This cell is put inside a solution that has the same amount of water as inside the cell. So everything is equal. They're isotonic. Iso kind of means equal. Okay, same concentration. There's, maybe an equal flow in and out, but it stays in this constant condition. This is where most cells want to be in. Okay, if we're talking about things like red blood cells, they want to be in an isotonic solution where things can just pass comfortably. Okay, the same amount of water on the outside here as the same amount of water on the inside. Hypertonic solutions mean that there's a higher concentration of dissolved materials outside the cell. So that means there's, think about if this cell is put in salt water. That means there's more salt on the outside here, okay? That means there's the water concentration on the inside of the cell is greater. We always know water is going to move from a higher concentration to a lower concentration, so water is going to move out into this hypertonic solution. Hyper means like greater, so it's got a greater amount of dissolved substances. If I said salt water here, there's more salt in here than there is inside the cell. So there's a greater concentration of water right here in this cell. Water, of course, is going to move from higher concentration to lower concentration, causing it to move out, trying to fill in these this hypertonic solution, that cell is going to actually start to shrink because it's losing water volume. Hypotonic solution, that means the liquid on the outside of the cell, or the fluid on the outside of the cell, has a lower concentration of dissolved materials. So say it's less salty, in just an example's sake, than the cell itself. So that means there's a higher concentration of water actually on the outside because the dissolved substances are less. Hypotonic, that's what the solution is described as. So more water concentration on the outside, less concentration on the inside of the cell. Water is always going to move from higher concentration to lower concentration. It comes in here trying to equalize, stabilize between the outside and the inside. It starts to swell up, eventually could burst. Okay, and that's all because they're trying to get to this isotonic equilibrium, if you will. So it depends on the solution that certain cells are put in. Ideally, say inside our body, things are isotonic. Okay, and cells are able to move things passively, through the cell membrane here and there with no big issues. These solutions give issues. Obviously, we don't want our cells shrinking up and dying or swelling up and bursting. Okay, you can come back to this slide as you need to. Just another, illustrative purposes here. Hypotonic solution. It's got a less concentration of dissolved materials, more water on the outside, more pure water on the outside. Water rushes into the cell because it's moving from higher concentration to lower concentration, burst. This is good. Equilibrium's reached in isotonic. Hypertonic solution, more salt and things on the outside of the cell, or more dissolved substances, which means there's a concentration greater of water inside the cell. It rushes out because water always moves from higher concentration to lower concentration, causing the cell to shrink up. Okay. So facilitated diffusion is another form of passive transport. Okay, it's another form of passive transport. When we looked at the channels, we mentioned these channels inside the cell membrane. Sometimes larger molecules need to get through there. This is the doorway. Okay, it just moves from a higher concentration to lower concentration, but it allows it to pass through these little channels. That's all facilitated diffusion is. So instead of moving in and out of the membrane from a higher concentration to lower concentration, it kind of takes that special door. Nothing really more. Biggest thing is facilitated diffusion is passive, requires no energy. All right, so let's talk about what happens when there is energy required. Passive energy, no, there's no energy. It's moving from a higher concentration of whatever molecule we're talking about to lower concentration. If that's water or, anything else, sodium, things like that. But active is kind of going against the grain. And that's why it requires energy. It requires energy to move things in and out. This is going against the concentration gradient. So higher to lower makes sense. It's moving from higher, rushing into a lower concentration. This is going against that. See, just like in this example, it's trying to move it from a lower concentration to a higher. And it uses energy in the form of what's called an ATP molecule. This is adenosine triphosphate. We will learn tons more about adenosine triphosphate. Just know that's energy inside the cell. Active transport requires energy, just like being an active person requires energy. Several different types we can look at. Endocytosis is the movement of liquids or large molecules into. It kind of sounds like it. Into. It moves them by engulfing them in the membrane. There's a special type of endocytosis called phagocytosis, which means cell eating. Really cool. White blood cells. We'll look at a video right here in a second of phagocytosis. But let's think about how it goes against the grain. So we're going against the concentration gradient. We've got this food molecule or this food particle, whatever it is, moving against the cell. What happens is it bumps into the cell membrane. Okay, it bumps into it, starts to form this little dimple, right? It forms this pocket that's surrounding it. Remember, this membrane is it's very flexible. The fluid mosaic model shows it kind of bends and twists every which direction. It's very fluid. So it bumps against it. Eventually, it starts bumping enough that it forms in. It looks like Pac-Man right there. It's moving in, and bloop, this membrane starts to seal up around that particle. Okay, and now it's been engulfed. A vacuole forms around this membrane or this particle. It will then take it wherever it needs to go in the cell. It'll get broken down or used for some type of energy in the inside the cell, but it's gone against the concentration gradient. This food particle, if there was more of these out here than there were inside, it may just flow freely through. That's diffusion, right? Or facilitated diffusion if it used a channel. But let's say there's less concentration here than there is on the inside. It's moving against that concentration gradient, and so it has to make it like this. This is when something moves into the cell. Phagocytosis, I told you this is a special way. This is an awesome video. Let's speed up. This is bacteria here. This is how like immune cells work. There's just some music playing over the top of it. Look how it just engulfs this thing. Look how this is it coming up, and it starts to eat, if you will. This is just some cutesy stuff moving through it. When we find a foreign invader, you know, it wants to come eat it. Now watch this. This is the bacteria right here, and this is the cell coming, a white blood cell, and it's whoa, it jumps out there. Did you see that? Like how it like attacks it, whoa, to eat it. So that's really cool. Phagocytosis, instead of making that little dimple like we saw with regular endocytosis, it actually goes out and gets it. I love it. Exocytosis is the opposite of endo. So exo, kind of like making an exit. This is when it releases substances out of the cell. Vesicles move the cell membrane up to the cell. From there, it kind of attaches onto the cell and then releases it. It's just the right opposite of what's happening here. So think about it starting at this side. It's moving something out. It kind of attaches here. A opening starts to form. This part of the cell opens up. The cell membrane opens up, and then we release that food particle or whatever it is outside, and then the cell membrane returns back to a normal condition because it's very fluid. So endocytosis is how it moves into it. Exo is the opposite. It's how it releases. It's how it goes back. So you would think of it starting right here. It forms this vesicle around it. It moves it to the edge of the cell. It attaches to the edge of the cell membrane, and then it releases us out. Okay, and all of that, the endocytosis, another form of phagocytosis, which is just a form of endo, and then exocytosis are active transports. All right, they're active, meaning they require energy. The biggest difference, hear me out, the biggest difference between active transport and passive transport is active requires energy. Passive doesn't require any energy. It's just simply moving from higher concentration to lower concentration. Please watch that other video that I posted. I think it's called Active versus Passive Transport. Check that out. I know you got a writing assignment for me, and also you need these notes. So you might want to go back through this slideshow and catch it again. Okay? Email me if you got questions.